Hi 11s and 12s. This is our fourth video in the DNA profiling PowerPoint this week. 2 more videos to go after this. They are shorter though, as you've noticed. In this video, we're going to be looking at DNA sequencing. So how do we actually physically sequence DNA? First off, I suppose we probably need to discuss what DNA sequencing actually is. It's the process of determining the nucleotide base sequence of a DNA molecule. So it's great that we know the size of the DNA fragments and we can identify if someone was at a crime scene. But when we get to genetic engineering, etc., we need to physically know the exact nucleotide base sequence. And that also helps with evolutionary comparisons, knowing how close or similar or different DNA is between different organisms. There's a number of different processes used in DNA sequencing. So first off, gel electrophoresis is used again. We use this in the chain termination method. Now, the chain termination method uses PCR as well as modified DNA molecules. Now, remember before we called the DNA nucleotides, we called them DNTPs. These modified versions are called ddNTPs. Okay, and what's special about them is they can't physically form sugar phosphate backbones or even sugar phosphate bonds. Okay, that means when you get to a ddNTP, the creation of a new DNA molecule stops. Okay, and therefore we know wherever it stopped, whatever ddNTP we're using, so maybe it's a ddNTP for adenine, we know if it stops there, then that was the last adenine needed. Okay, so how does this all work with DNA sequencing? I'm glad you asked, because the actual method for DNA sequencing, okay, we have 4 different test tubes, and we fill them with as many copies of the target DNA molecule as we need. So many copies. We then, into each test tube, we add all 4 of the non-modified nucleotide bases, so our DNTPs. So we've got adenine, cytosine, guanine, thymine. Okay, and then into each test tube, we also add one of the modified one type of the modified version. So in test tube 1, we might have ddNTPs for adenine. Test tube 2, we might have the ddNTPs for cytosine. Okay, for example. And there's more than one of them put in there, but only one type in each test tube. Okay. We also add to each test tube primers to help the elongation of the DNA chain. We also add DNA polymerase enzymes to help create the sugar phosphate backbone and place the nucleotides in place. Okay. We then place all 4 test tubes into a thermocycler, and we let that process happen. Now, DNA replication will stop whenever that ddNTP is added. Okay, because no other ddNTP or DNTP can bond with it. So at the end of that, we can remove all the different DNA fragments that we have from the 4 test tubes, and we can run them through gel electrophoresis. And every time we look at a different size molecule, whatever one that test tube came from, so say we've got one molecule that's got 4 nucleotides on there, we know the 4th nucleotide has to be adenine because it's got the ddNTP for adenine on it. And we do that and look at all the different size molecules so that we can figure out which the entire sequence of the nucleotide in that DNA molecule. Okay. We also use something called electropherograms for this, and we'll talk about electropherograms a lot more in video 6. Okay, but a little bit of a rundown of what's happening. So fluorescent chemical compounds are also added to our ddNTPs. Okay. That means that at the very end of the fragment, we've got our fluorescent ddNTP molecules that are able to be seen under UV light. Okay, we use our gel electrophoresis to physically separate those fragments by size. Generally, it will happen inside capillary tubes in a DNA sequencer. Okay, you don't even know what that looks like or what that actually is, just that statement there. It takes place in capillary tubes of a DNA sequencer. And then because of gel electrophoresis and the different size molecules, the DNA fragments will go through those capillary tubes at different rates, with the smaller particles moving faster, just like we see in gel electrophoresis. Okay. Now all of those fragments will then pass through a beam of laser light, which will be absorbed by the fluorescent molecule. Okay. We then put the whole system of fragments through a detector, converts those light signals into electrical currents, producing a graph called an electropherogram, and we'll look at that in video 6. Okay, each peak will represent one of the different nucleotides with the height of the peak indicating which one. And we'll do some questions this week on this. Okay, so you physically will have to be able to determine the sequence of DNA. We'll do some questions on that this week. But for now, just knowing the process that an electropharogram is created with.